And I think the starting gun is basically 2022. And what we're going to see, I believe, if people put the effort in, is a lot of structural biologists who know how to use these computational tools matching up with experts in particular fields who know a lot about diagnostics, or who know a lot about the heart, or who know a lot about a given infectious disease, or know a lot about a given agricultural problem. And in combination, I think those teams of people are going to do really incredible things.
All right. Well, we've talked about proteins, all the cool stuff proteins can do. We've talked about the history of insulin, one of the protein treatments used in treating diabetes. We then talked about improving proteins with AI to be used in medicine. And now I want to hear from you about how we can design entirely new proteins that have never been seen in nature. But first, why would we want to do that?
Nature's great, and I've got nothing against it.
Mm-hmm.
But
I have some things against it.
Okay.
Like, I don't know, natural disasters, tornadoes,
Tooth and claw.
mosquitoes.
Yeah, mosquitoes, gosh. Some of them, I guess, are harmless, but some of the others. Okay, nature's not perfect, but nature's so good when there's a problem that evolution's really taken a swing at. But there are many problems that face us as humans, as a society, as a planet, that nature's not in the same sense been evolving to try and solve. So, for example, plastic famously is not biodegradable.
Right.
So nature's not in the sense of bio, ain't doing much degrading.
So what if something could digest it?
Exactly. Could you design something with that problem in mind to try and digest it and get rid of all that plastic currently in the ocean?
Could we get a Pac-Man to eat all those little blobs?
There's also these sort of pathogens that we had talked about last episode, one of, but some of them we haven't been able yet to, either our body in terms of our immune system or drug development has not yet been able to make drugs that actually work well enough. Mm-hmm. If you could create a protein that operated as a therapeutic, as a drug, and you just create it out of thin air and you never saw anything like it before, then it could be really useful.
I think there are maybe also lots of uses outside of medicine. I recently learned that silk is a protein.
Oh.
And we talked about gluten in bread. That is a protein. So there are all these proteins that are also doing these like structural things. Right. They're stretchy, or they're super strong, or they're like really silky and smooth. And maybe for other purposes like materials or industry, you could want lots of proteins. And then maybe there are other things in like industry as well. Like maybe you want to cook something, you want to like ferment something in a way that's never been done before. Mm-hmm. You might want a protein. You might want an enzyme to do that for you.
Yeah.
And then what else? Like maybe there are just a bunch of chemical reactions that you want to do.
Oh, totally.
But at a really small scale. And you want to do them all at once. And you want a protein to do all of those steps of the enzyme reaction. And you could create a new protein. Or maybe you want to create a protein that does like a bunch of different things. And like a protein that's like, okay, in a hot environment, it's going to do one thing. Cold environment, it's going to do something else. That like tunes the protein and what it's working on.
I also want to just get energy. I mean, this is...
To eat,
to do cool stuff with like photosynthesis. Mm-hmm. One of the main
you want to do photosynthesis.
I want to do photosynthesis.
Would that turn you green?
By the time I succeed, I may have worse problems. I just feel like photosynthesis, I don't mean to criticize plants, but I've always found it very inefficient. You've got so much sunlight beating down. Hardly any of it gets turned into biomass. Well, like 1%. I can't. Oh yeah, hardly any. Any. And it's probably some deep reasons for that. But could we design new proteins, not just like Rubisco that you mentioned in the first episode, but even better ones?
Right. So proteins are used to get carbon from the air. They're also used to get nitrogen from the air, right? To fix nitrogen. Right. And so you could be trying to improve some of these processes to get stuff from the air.
Yeah.
You could be trying to improve agricultural yields or like
Yeah.
products that we eat. What else? Maybe you would be trying to purify water, trying to make new biofuels.
Oh wow, purify water.
Oh, I feel like there's just so many things. Yeah, purify water. You could like get rid of, maybe the proteins are really specific and they attach to dirt or something. And they eat them up.
Okay. I eat your dirt. I eat it up. I think that everything we just described sounds wonderful, but sounds a bit magical. So how would you actually go about achieving a protein if you're not modeling it off of something in nature?
Well, a lot of the things we described actually happen. They already happen in nature.
That's a good
Maybe
point.
we just want to adapt how they happen. We want to make the silk stronger, or we want to make the gluten stretchier, or we want to make the silk stretchy and the gluten stronger.
Or we want to make the dirt dirtier and the water. Fair enough. Fair enough. I think we still need a way to invent these proteins, though.
Mm-hmm. And maybe you want to make stuff that exists but is not protein, but you want to make it protein. You want to make it biodegradable or something.
Yeah.
Like, I don't know, you want a flower vase that is made of proteins or something. And so you could probably make things like that with proteins if you wanted.
I want to tell you about the next thing I did when I visited the University of Washington.
When did you visit the University of Washington? What were you doing there?
I was there in March, and in the last episode, I talked about how I was working on a protein on the strep A bacteria. I want to talk to you about my next project, which was designing a drug against a hepatitis B protein.
Ooh, wait. So you're designing a drug, not a protein?
Well, the protein is a drug. No, the drug is a protein.
What?
I know. By drug, I just mean in this context, a binder, something that binds really tightly.
Okay.
So before, in the last example, I was talking about taking an existing protein that exists in nature and tweaking it a bit. Here I'm talking about hallucinating an entirely new protein that has no previous instantiation necessarily in the world, using just a diffusion model.
Ooh.
So you've seen DALL-E made by OpenAI. You've seen.
I have made some cartoons with it.
Nice. What about stable diffusion and?
No.
What about
Midjourney?
Thank you, Midjourney. Oh yeah. What about Midjourney? I mean those have gotten really good these days. So imagine, I mean, I'm speaking a bit loosely here, but not that loosely, that instead of hallucinating cat pictures, you started hallucinating a protein structure.
When I was in school and I was learning how to play the piano, and at the end of school, when I was like 16 or so, well, I was doing the grades of piano, like qualifying for them. And there was this one exam that you had to do where you had to play the scales, regardless of where the examiner told you to start.
Oh, okay.
And I remember being really bad at that because, like, you have to remember where your fingers go, like which order, do you use your third finger at this point, or do you switch back to your thumb? And I got so stressed out by this whole situation that I would literally start dreaming of myself playing the scales at different like points. And it was it was genuinely helpful.
Oh,
yeah.
Hallucinations can be helpful. And you can actually use a few AI systems in a design, create, and validation loop to get to new proteins that might be useful. So
wait. What does this mean? So, okay, you said that you're making a binder. So you are trying to make a protein that sticks to another protein to do what?
In this case, so lots of functions begin by sticking. Let me just say that to begin with. But in this case, the reason I want to stick is that I want to bind a part of hepatitis B to interrupt its life cycle. So it stops damn replicating in my liver cells.
You're like, shut up.
Stop. Yes. So actually, I went after a sort of little discussed protein called...
What? Wait, what is a disgust protein? What did you just say?
You know, everyone talks about the hepatitis B surface antigen. I've never heard anyone shut up.
I'm sorry, I've never heard someone talk about this. I know about it because it's used in the malaria vaccine, right?
Oh, that's absolutely true.
But
yes.
Why is everyone talking about this around you?
Okay, let's do a quick side note on the... Okay, firstly, I love that you tried to say you don't hear people talk about it, and literally you talk about it all the time.
I read about it. I read about it. I've never heard someone talk about it.
Well, now's the moment.
I have normal friends.
I know for a fact that is not true. Especially given I am one of them. Okay, so let's do a detour on the malaria vaccine because that's fascinating.
Right.
The original idea is you take the hepatitis B antigen, surface antigen, which happens to self-assemble into this kind of, spherical thing.
Yeah.
And that's pretty useful because, the immune system's good at looking at spherical things and being like, that's a virus, kill it.
Mm-hmm.
And if you can lace that spherical thing with antigens from the malaria parasite,
right,
then inject those.
Yeah.
Oh boy, we're in business.
Yeah.
Yeah. That's a cool idea.
Right. But it's like if I was trying to detect you and your face, even if you were wearing a wig, I should still be able to recognize you because that's really important.
Right. I like how most of our metaphors end up with you assassinating me. So, hepatitis B surface antigen, it's the rage. Everyone is always on about it, as we've now fully agreed. And the issue is, the drugs for hepatitis B are pretty good in that you definitely want to know if you have a chronic infection because you can go on nucleoside analogs. They will help you control the infection. They won't cure you, though. So the drugs aren't good enough to cure you yet, although many people are working on that. So I was kind of like, let's go for the jugular. What can we do to really get rid of this thing? The hypothesis is that if you create a protein drug that binds really tightly to this other antigen that's actually in the middle of the life cycle, which is called the X antigen.
All right.
And
this is very scary.
It's very scary. But the X antigen, it forms as two parts, a dimer, that come together and then start doing stuff. So I was like, okay, I'm going to hallucinate a protein that interrupts that dimer formation. Can I distract it and get it to bind?
Right. So you're imagining what might fit into the little gap between these two parts of the protein.
Yeah. And I used this tool, RFDiffusion, which is a particular diffusion model made, again, at the Baker Lab at the Institute for Protein Design. There are other diffusion models being made too. The RF there stands for Rosetta Fold, which is the kind of family of models that they've worked on up there for a while. What I'm doing there, I'm saying, giving the computer just a few inputs of what I'm attempting to do, asking it to hallucinate many options.
I saw a video about this, and it featured a lot of cats. And I think you mentioned some of these cats before.
Right.
And it's a bit like DALL-E. And again, I didn't even know how DALL-E worked, like how it was like developed and stuff. But the basic idea is you have a bunch of pictures of cats or something. And you introduce some noise into that image.
Right.
Or maybe you're making
pixelated.
Yeah, more pixelated. Or you just introduce random little pixels that are like different colors or something. And you introduce some of that, and then you do that again. You make it even more noisy, and then you make it even more noisy. And so you have like different versions of the same image that are like progressively noisier.
Yep.
And what you're trying to do is get the image model to try to figure out how to go backwards, how to get from the noisy version to the clearer version to the clearer version to the cat. Yeah. Meow. And then and then this was really funny because I was watching this video and, if you input a cat, you've made it noisier. And then you're telling the AI, okay, now try to predict what happened before. Like, what did it look like before I added this noise? And it's like slightly less noisy, and then it's a dog. And you're like, wait, that's not right. And you just keep doing this until it gets better and better at predicting what the image is.
Right.
And then what's happening with the protein version of that, which you described as RFDiffusion, is that instead of having an image with noise that's like the little pixels, you instead have the coordinates of the atoms in the protein. And then you introduce a little bit of jiggle, like you like mess up the coordinates a little bit. And I think in this case they add some Gaussian noise, so they
Right.
like move it by basically most of the time it moves by some average amount, but sometimes it moves a more extreme amount and stuff like that. And then each time you're making it blurrier and like messing it up more and more. And then you're asking the AI tool, RFDiffusion, can you go backwards and remake the protein? Or like what? And then obviously there are issues with doing that still. Like it's not going to be very accurate, but in this case, it could be a good thing that it's not accurate because you're creating whole new structures that you haven't seen before. And some of those structures might be useful for other purposes. Why would you want to hallucinate something that doesn't exist? Maybe there are just so many more potential ways that a protein could fit together that have never been seen in nature before.
Yeah.
And you're like, instead of a cat with one head, what would it look like if this cat had 3 heads or something?
Let me just describe the loop you can then go in. So what RFDiffusion, which was made by, again, this group at the Baker Lab and the Institute for Protein Design, Helen Eisenach and others made it. Well, you can generate these different hallucinated structures. Okay, so this is, again, think about like a 3D model of where is this protein backbone. What you don't have is the sequence. So you remember from last time that you can go from structure to sequence using ProteinMPNN.
Right.
So a different model.
Yeah.
Okay, so I'm going step 1, hallucinate. Step 2, okay, hold on. What sequences would actually lead to those hallucinations?
So what structure would fit in between the gap, and then how do we make that structure?
Exactly. And then you're going to want to do the validation or the first in silico step of validation with AlphaFold.
And that is basically, if I make this amino acid, does it actually make the structure that fits into the little gap?
Yes, I have these ideas for the amino acid sequences, but in reality, is it going to fold up to look like that?
Right.
And what you're going to end up with at the end of this 3-step chain is you're going to end up with some hypotheses, and AlphaFold's going to say, look, I'm sorry, some of these are not what you thought. Like, it's pretty unlikely that amino acid is the one that will lead to the thing you want. It's pretty unlikely that the distance between these 2 randomly selected amino acids is going to, after folding, actually be where you thought it was. So you want to ditch the ones that you actually accidentally messed up on the way. AlphaFold, AlphaFold, AlphaFold.
Yes.
We should probably explain a little bit about why that was a breakthrough, how that came about, what was actually happening with protein folding before.
So I think I described at some point the fact that people used to be using physical models to predict what protein structures were like. So sometimes they had the structure in mind and they were trying to figure out what amino acid sequence goes into that. How do the amino acids look like if we make them in physical structure, like with a model, with a real life model. And then I think after that were these statistical models that were produced with different types of information. So you might have some data about each amino acid, what kind of features it has, like how it interacts with other amino acids. Another thing that you would have is data on like the amino acid sequence for a particular protein, but in different organisms. So you might have, what does insulin look like in chimpanzees? What does insulin look like in pigs? Etc., etc.
Yep.
And then by comparing all of these versions of the same protein, the different amino acid sequences for the same protein in the different organisms, you can see which parts are basically shared, right? So you can see which bits are basically in the same are shared between all of them, and that tells you something about the important structures that are kept in the same shape. But it can also tell you something else. Like, it can also tell you if some parts of the structure are changing, do other parts of the sequence also change along with it? So you're like, okay, if leucine always changes here, and whenever leucine changes here, then alanine always changes there, or often changes there, or something. And with these like comparisons of different, of like having a pair of a comparison, can tell you a little bit. And the reason this is important is it means that they probably interact, that they're like probably close together in the 3D shape of the protein.
Right.
And that is useful information. And then another thing that you might know is you might have some information on secondary structures of proteins. And what that means is like in specific parts of the 3D shape, what is going on? Like, is there a little helix? Are there two parallel lines or something? What's like, and then how does that map onto the amino acid sequence? And so if you have a bunch of this information, you can try to predict what the structure would be like. And so there were people who were working on some of these models for a while, and they were making some gradual improvements.
Structural biologists.
Yeah. No. No? Because the structural biologists are they're determining the sequence by using crystallography or cryo-EM or something like that, right? Whereas what I'm saying is, can you predict it computationally?
You think those were different people in computer science departments? Or
maybe it was some of the same. Yeah, I mean, it's different tools. But when I think of structural biology, I usually think of the imaging and stuff.
Yeah.
Okay, but so people are making a little bit of progress, but the predictions are still not very good. And that continued for a long time. And also there's, there are people who are saying, oh, my model is really good. My model is better than yours. And there's no benchmark or there's no reference to compare them.
Yeah.
And so what happened was in 1994, is that right? At UC Davis, people developed a competition and they said, okay, some crystallographers or like some people have actually figured out what the structures of these previously unknown proteins looks like. And we're not going to tell you what that structure looks like.
Okay, but that's real. That's documented.
Yeah, that's like determined in the lab.
Prediction.
But we will tell you the sequence of amino acids, and you have to guess what the shape is like. And so now you have this very standard, comparison where you can give all of the different research groups this amino acid sequence and say, hey, can you guess what it looks like? And so they came together for this competition, CASP, that was set up in 1994. And they were given a bunch of amino acid sequence and told to predict the structure. And then you could compare how good their predictions were.
Yeah, it's the real thing.
And so I think until, what is it, like decades, okay, there are some gradual improvements, but basically the accuracy is under 50% generally on average. And that accuracy is about, how far away are the coordinates that people are predicting of the atoms to the real structure. And then in 2020, DeepMind released AlphaFold 2, which was this model that not only used that data, but it also used structural data from a data set called Protein Data Bank. So people had already determined this structure from, X-ray studies and blah, blah, blah. And they had collected lots of this data on like, if we have this amino acid sequence, this is what the protein structure looks like. And they had done this for hundreds of thousands of proteins, right?
Yeah.
And AlphaFold was trained on all of this data. So it has a connection directly between the amino acid sequence and the structure. And that means that they were able to make a much better prediction. And their prediction was so much higher. It was like around 90% accuracy on average for the particular proteins that they were asked to determine. And, there's still stuff that it can't predict. even now, and there was still stuff that I couldn't predict then. But that was like a huge leap.
That's like a game changer, yeah.
Yeah.
Because if you can go from the number of proteins that us mere human beings had crystallized over the last 50 years, so maybe that's 200,000,
Right.
and you suddenly have a tool that can predict pretty well a lot of proteins. I mean, you can predict how millions of proteins fold without having to crystallize them.
Mhm.
You're not totally sure, but it gets you so far.
Right. And it's doing better on proteins that look similar to other proteins that people have determined in the lab. And it's doing worse on ones where there's like hardly anything to go on. And then also in particular areas of the protein, some domains are going to be determined quite well because there's lots of data on them already. Some are going to be predicted much worse. But yeah, that's the story.
It reminds me a bit of these other parts of machine learning before the deep learning revolution. Like you had, well, I don't know if I'd even phrase it that way, but before they just shove loads of compute at it revolution, where, you had these computer vision grad students who were writing these algorithms for edge detection and, trying to understand what's in an image with all these clever algorithms. And then sure enough, you just sweep through with a ton of compute and a particular architecture, and you can learn all that stuff without having to yourself get that specialized about it. And I imagine a lot of people who are working on these physics-based models, these other models before AlphaFold, they probably are quite intricate, are a bit like, oh my gosh, what? I was working so hard on this subset of proteins, on this type of, like when alpha helices look like this together, all of that. And lo and behold, I got blown out of the water by a bigger machine.
I think it's also, maybe at the start when people are building these physics-based models, they have this like textbook understanding of some parts of the process. They have some, I guess, like training and expertise over many years that they've learned of like what might fit together and stuff like that. But it's really hard to consider how all of that works on a grander scale on like a larger protein. Especially when it's like types of proteins that you might not have come across. And obviously there's hundreds of thousands, there's so many types of proteins, right? And so even when you have someone with the expertise, going from that to a statistical model can add value. And that was what came before AlphaFold. And then AlphaFold is also, in a way, it is a statistical model where it's like learning from some of this data and it's predicting things better. And part of the reason is not only does it have that information about like particular amino acids, but it also has lots of structures that it's like remembered. And that's very hard for a person to do, or a statistical model.
A puny statistical model.
Right. Yep. So let me do a quick recap of all of the tools just to see if I remembered them right.
Great.
So we first have AlphaFold. That's maybe the most famous one that people would know about. And that does, you have the amino acid sequence and you're asking, what structure does this make if it was a protein?
Yep.
And then you have ProteinMPNN, which is the opposite. It's like, I have the structure. What amino acid sequence makes that structure?
Exactly.
And then you have, thirdly, RFDiffusion, which is like, given that I have some bits of the structure, can you make the full, or like, I want this protein to have these things. Can you make the full thing for me? And like, can you make a full thing that could be a protein?
Yeah.
But then you need to check that that happens. Like, does the protein actually fold that way? What is the amino acid sequence that makes that protein? And and if you had that amino acid sequence, does it actually fold into that protein? And the reason that I guess that's really important is because there are so many potential combinations, right? Like, there are 20 different amino acids. So at any part of the chain, there could be one of 20 things. And if you add those up, I think if you get like a protein that is like 60 something amino acids long, the number of combinations is already bigger than the number of atoms in the universe. That's like the estimate. which is a huge amount. And like,
Yep.
so you want to make sure, is that the right structure? And is this structure going to fold into that particular shape? And that's also hard because there are many potential ways that a protein could be folding.
Mm-hmm.
Yeah, because there are so many different amino acids at different places, it could just fold into a different shape or conformation. But I think there's also this question of like, what is the structure that it will fold into, given that it wants to reduce the amount of energy that's required to like keep in that position. So it kind of wants to go down the easy route, but then there might be many easy routes, right? And there might be many multiple different ways that a protein folds.
Yep.
But in reality, it usually only folds a particular way, I think.
Right.
Yeah. So, okay. So that was my recap.
That's a great recap. And I think the real practical output of what you're saying about the combinatorics, it's, it used to be the case if you had hypothesis molecules, whether they were protein binders or small molecules or something, that you were trying to achieve some function. So you were trying to bind this hepatitis B thing or whatever. You might have to run through high throughput screening. You might have to have 100,000 different miniature experiments of 100,000 different hypothesis molecules, because most just will not do what you want. And what's astonishing about this loop of 3 systems that we just described is that for proteins, at least, you can go through the loop, do the down selection on your computer. And once you've got it set up and running, like I literally did that in a day. Like, once it's set up and running, it was based off of not only all the work of people building those models, but also being next to extremely helpful other postdocs and grad students, but who would, share their Jupyter notebooks with me and show me how to do it. But, you can create hypotheses, some of which do work, and you only need tens of them. Okay. So they work for the initial in-lab validation step. I mean, they don't necessarily work as actual drugs in the field once you go through all the future steps of getting through humans, but you don't necessarily need 100,000 things. You could actually try 50 and maybe 4 will work. Yeah. And that is completely different than it was literally 5 years ago.
Right. That's crazy.
Crazy.
I have 2 thoughts or one question. One is I was thinking about, I think you mentioned something about like the errors and like how well it's predicting stuff. And I guess is that with AlphaFold, I think? So there's like, you're predicting the structure from the amino acid sequence. And then the question is like, how well does this map onto what the protein actually looks like? And what is its real structure like? And so what you're comparing is like at each particular atom, even.
Yeah.
How far away is that in coordinates to the atom in the real structure?
Well, yes. My understanding is that what AlphaFold is giving you is its own, if you will, subjective predicted error versus reality. So you're not actually ground truthing a lot of it. Okay. But you could. You could, in theory. Well, if you're able to crystallize a protein, you can do that. But some proteins you simply can't do it.
Right.
But yeah.
And so you have confidence or like error or something, but that is not just of the shape as a whole. It's also about at each atom.
Yes, exactly.
How confident is AlphaFold that it's got that position right?
Yep.
And I think that's really interesting because it's like, maybe there are certain domains of a protein or something like that where we have loads more data about this type of protein or this part of the protein structure. And so AlphaFold can be much more confident that it's going to look like this. But in other parts, they might be completely new, or just like the structure hasn't been determined by anyone before in similar proteins. So it doesn't have that much to go on.
Yeah.
And it's kind of just like, I don't know. But there's the other question. Is it possible to make this protein in the lab? And maybe there are other challenges in doing that aside from just whether it would fold into that shape. Or would the protein do the thing that you want it to do in the lab or in real life?
And it's going to depend on the protein. I mean, the difficulty in making the protein, it is more difficult still to make large proteins, for example. It gets more expensive. You got to splice together multiple things, all that. Will it do what you're looking for it to do? You know, it's all about what is the initial test that you can do to get some signal.
Right.
And that will be different for different things you're trying to achieve.
And I think maybe there's also differences because in, let's say, protein data bank, or like the structures that are being predicted are just one static version of the protein. They're not like,
yes, absolutely,
a protein in real life is wiggling, or it's like moving around, or it's like folding, or it's turning and rotating and stuff like that. And that is not being predicted.
Nope.
It's not predicting how it binds to metals or something like that. There are some other tools that do that, though, that are kind of based on this. But then you would have to have data on like what that looks like as well.
It's good maybe just to summarize the useful things that aren't yet done. So there's that, what you just said is completely right. We just don't have good predictions of protein dynamics. So we're pretty good at predicting protein structure now. But as you said a second ago, not perfect, but we're getting there. We can't yet predict protein function very well. And so I just want to distinguish what we were just talking about is starting with an intended function, hallucinating a protein to serve it.
Right.
That was very difficult 5 years ago and is increasingly some problems you can do that for. So that's start with a function, hallucinate your way to the finish line. But we can't, if you just take a given human protein, it probably will serve multiple functions. And you're not going to suddenly be able to ask an AI, wait, what is this protein doing, by the way?
Yeah.
Like that is, that's one of the holy grails left, which is, we care about function more than structure, ultimately. Right. So,
and I guess the other thing is maybe also, is it attached to something? Is it a protein that's stuck to a membrane? Does that change its shape or something like that? And that's not something it's answering.
Yep.
But it's still really useful because a lot of proteins are kind of just hanging around. They're just like dissolved in something. Yeah. And they do sometimes look like those structures.
Yeah. And, let me give a shout out to a fourth AI model.
Oh.
Don't know if you heard of ChatGPT.
Uh-huh.
Claude.
I
Gemini.
have heard of those.
I was talking to a grad student who was at the frontier of these biological machine learning models. And she was emphasizing to me how useful ChatGPT was, because this loop with 3 models we just discussed, it's really simple, but you do have to know how to code to get it to work. And simply learning to code takes a while.
Right. What language do they use? Do you know?
In my, I'm sure there's many answers. That Python is classic that you would start with, maybe. And if you're a grad student and have to take a year out in order to learn to code, just so that you can use these models, that's what it used to be like all the way back when these models were 2023. So these models were out, but we didn't yet have really good LLMs. I mean, luckily in 2025, you no longer have to take a year out because you can basically talk to a large language model about like, okay, what's this bit of code doing? Okay, write me some code that does this. Now explain to me what that function is doing. Okay, now explain to me what I'm missing. And it's
your vibe protein designing.
Vibe.
I have sometimes used ChatGPT to like code stuff, but it's like, it's kind of hard because when it makes a mistake and there's like some error and I run the code and then I say, hey, you made this mistake, or this was the error. And then it's like, it finds it really hard to fix the error. Like, it's like, it doesn't know where the bug is coming from. But then I also use it sometimes because I share all of my data and graphs on GitHub.
Very good.
I have a background of using R, the programming language, but I don't use Python.
Okay.
But I think that people who use Python should also be able to reproduce my graphs. So sometimes I have written my code in R and I tell ChatGPT, turn this into Python so that someone else can just run the code super easily. And then again, like, I don't use Python, so it's like hard for me to fix any issues that come up. But I will like copy that code and then run it on the terminal and see if it makes my graph again.
Yeah.
And it does take a while, but like it does eventually get there, usually.
Yeah.
Which is cool.
That's great. Another one that brings to mind for me is, you're often using other people's code or inheriting some of other people's code when you're trying to achieve something, and different people document their code well or poorly. And someone who has not done a good job of factoring their code or documenting it well, you can at least ask one of your friendly AI assistants, what the hell is going on here?
Yeah, that's helpful.
Yep.
But it's like, it is helpful for the lines of codes. And then when it's a very long script, it's just like, I've forgotten what I'm doing. I'm sorry.
yeah.
But in fairness, I would forget as well. So,
by the way, shout out to Sebastian Ols, who has famously well documented code for some of the systems that
Sebastian Owl?
Ols, O-L-S. I don't know how to pronounce that Swedish name.
Interesting.
Yeah.
Well, thank you to him.
Thank you to him.
Where were we with hepatitis B? I've like almost totally forgotten about it.
Well, I actually have a video I can show you of how far I got that I took on the final day while I was up there in Washington.
So wait. You were making a protein that fits into the gap between hepatitis B's proteins, and then you were like, how do I block them from joining together?
Exactly.
And you used RFDiffusion to hallucinate a potential protein that would fit into this gap. And then you went, what amino acid sequence creates that protein with ProteinMPNN? Yep. And then you asked AlphaFold, does this amino acid sequence actually produce the structure that I want?
Absolutely right.
I remembered.
And by the end of that system, I've got it down to 50, 100 different possible sequences and structures.
Oh, that's a lot.
It's quite a lot. I mean, you could go down further if you want, but the really fun thing is you can visually look at those structures in the thing I used was PyFol molecule. And you can see, does it look like it would line up and bind? And
this is a software where it shows you what proteins look like.
You can twist them around.
Represented with ribbons and arrows and blobs.
Exactly.
So in the graph we're showing, they're basically like each of the little dots is a predicted structure that you made. And the graph is showing what is the potential error in the structure compared to the subjective, you said, reference or something. Or like
basically
how far is this structure in terms of the coordinates and stuff from what it should be.
Yeah.
And because you've made so many of these predicted structures, you then filtered down and you went like, these are probably the ones that are going to look actually like this.
Mm-hmm.
And then what did you do next?
And then, those, I went home, to be honest.
Well, that's boring.
I know. But I love to leave things unfinished. What one would do next if I were a full-time lab scientist.
Oh, wait, you didn't just go home that day. That was the end of your time there.
That was my final day there.
Oh, okay.
So I hope that other people are carrying forward ideas of that sort, but those particular binders disappeared into the ether.
Right.
That said, what I could have done was order up the DNA sequences that would code for those proteins, grow up the proteins in some system, harvest those proteins, and check against hepatitis B virus or something like hepatitis B virus, maybe the protein itself. So you would be seeing, does this protein actually block it from binding to each other, to the two parts? And probably what I would find, given the state of the AI models, is that most of the things I ordered wouldn't, and some of them would.
Okay.
And I just can't emphasize how astonishing that last part is, because you used to come up with sequences and you used to make up proteins and they didn't work.
Right.
And now some of them seem to work.
Yeah. I mean, it might have been like a 3 or 5 year project just to work on one of these things.
And what I hope we'll talk about next is why even all of this magic sometimes is not enough.
I'm also wondering, okay, you mentioned binders. What are the other potential uses that you might have for this technology of like hallucinating different proteins? Could you, I don't know, is it like Lego? Can you make little bits of the proteins and stick them together? Would people do that?
That's one way to find out. Let's try. I mean, I don't know the answer, but maybe.
And I think I've seen some of these structures where they're just some symmetrical thing. Maybe they're like a tube or a ring, or there's some star-shaped thingy, and they're proteins.
Yeah.
And people have figured out how to make those. And I think that's maybe easier than making an actual protein that's doing reactions. If you know, for example, in a protein, this is how you make a little helix in one part, and this is how you make a little fold, and this is how you make some parallel structures. The computer can have a pretty good idea of putting that together and making some symmetrical thingy with it.
Yeah, you know who's done really cool work on this?
Who?
Chelsea Freeze.
Who's that?
Postdoc in the Neil King lab.
Mm-hmm.
And last time I saw her, maybe Fries.
How
Chelsea
do you
Fries.
spell her? Yeah.
Yeah, spelled like fries.
It's not like Frozen Freeze.
No, although ironically, she does a lot of cryo-electron microscopy.
Oh.
So maybe that's nominative determinism. But she is down there in the lab. Well, in the basement with the microscopes. And I went to visit her once, and she went, "Hey, Jacob, you want to take a look at this?" And on the screen, she had this perfectly symmetrical long tube.
Ooh.
I was like, "What the heck is that?" She was like, "This is a self-assembling massive protein tube."
That's so cool.
And I was like, "Okay, what? That's amazing. So what can you use it for?" And she said, "Oh, I've got no idea." And she is just has an instinct. If she follows this further, having nanotubes will probably be useful for something.
I can imagine tubes being useful for various things.
Yeah.
Maybe as a straw.
Right.
For little bacteria or something.
Yes.
I don't know.
For those little, what are those critters that, do you know who I'm thinking of?
What the hell are you talking about?
What are those? What are they called? Tetramegaphon, pterodactyls. The ones that look really ugly. Say again? Tardigrades.
Oh, tardigrades.
You must know tardigrades.
Well, tetrahedral what?
So I think that tardigrades need straws because currently they don't get Diet Coke in the right quantity.
Oh.
Yeah.
But do we want them to drink more?
Oh, they might get really frenetic.
Uh-huh.
Yeah.
But also I think we talked about this earlier when we were talking about our favorite proteins, and I said microtubules, and they're a type of tube. And they are used for the structure of a cell, the skeleton of a cell.
Wow.
And so I feel like there could be lots of cool uses for this.
Yeah, Chelsea's on
like scaffolding something.
Yeah, totally. Well, you could create a mansion, but the mansion is microscopic.
Like a little dollhouse.
And columns.
Yeah. For little end proteins to live in.
Yeah.
I think I'd enjoy that. Maybe that's something people would do with like 3D printing. They could like 3D print a protein, make a little protein house.
All a protein wants is a little protein house and a white picket protein fence.
A what?
A white picket protein fence. Like a white picket fence.
Well, this protein is going to be a NIMBY.
No. Okay, don't make NIMBY protein. That's our one request.
Okay, so we can think about hallucinating proteins for binders.
Yes.
To block binding or to make things bind.
Yes.
We could make scaffolds.
Yes.
We could make little structures of their own. Maybe the shape of the structure itself does something.
Yes, I think so.
Oh, hemoglobin, right, is a protein complex with 4 heme things. And they fit together and the oxygen fits inside them. And if it's just one heme, it can't carry the oxygen and let go of it, I think. And so maybe it's a similar sort of thing. Like if you can make a structure that does something, but in a complex it can do something else, and so you want to create a larger structure.
Right.
And so what happened with so you would have ordered the DNA and made the amino acid sequence, and you would have made the protein, and then you would have seen, does this actually block hepatitis B in the lab, maybe in some animals or something, and
Yep.
then in humans.
Yep.
So that's it. We're done.
If only, Saloni.
Oh,
We talked through one example of trying to make one hallucinated protein, and we hypothesized other possible uses. Do you know, have these proteins actually made it into the real world yet? And what have people worked on already?
I mean, this is a super new method,
Yeah.
right? It was the first time it was published was 3 years ago, right? In 2022. And so it is fairly new. I think there are a few proteins that are in the pipeline as drugs that have been developed based on methods like this or this method. One of them is called rentosertib, and that is a small molecule drug that is developed to treat pulmonary fibrosis. And that is a lung disease where, I think there are different things that can trigger that. But basically, the idea is your lungs have all of these little empty spaces called alveoli where your blood is kind of on the edge of that little space, and that gives it a little bit of a gap or structure where the oxygen and the carbon dioxide mix with the air, and that basically helps you breathe. And what happens with this disease is that there's some kind of injury or some inflammation or stress or infection or something that damages these parts. And then when the body is trying to repair it, it produces this like fibrous structure or this kind of repair structure, and that kind of goes too far. It's like the scar
Yeah.
that this scar is just a slightly different material than what was originally there. And that actually prevents the transfer of oxygen and carbon dioxide as well as before. And over time, for whatever reason, people's lungs get more and more scarred. And so they become less and less able to breathe well. And obviously, that's very harmful. And so this new drug is basically trying to block a particular protein that is involved in this whole process. So that's one that is in phase 2 trials right now. There's another one called luxdegalutamide, and that is used to potentially treat prostate cancer by targeting the androgen receptor that's involved. And that is also in phase 2 trials. There are a few that are in trials or were in trials but then got discontinued because they didn't work. And that kind of, highlights this thing that you were saying, that even after you've hallucinated a thing, you still need to make sure, does it actually produce this structure in the lab, but also does it do this function in the real world when we're using it as a treatment?
And does it avoid doing other functions that may cause
Right.
problems elsewhere in the body?
Yeah, is it causing side effects? And yeah, so I guess that's where we are now, but it's super new, right?
Yeah.
And so because clinical trials take so long, you wouldn't expect this to get to the market for a while. Do you have a favorite use of these tools or thing that you think they could do?
I am hopeful for several things. I'm just so curious to see how it all goes in the next few years. But one is actually proteases.
Mm-hmm.
So if you look at one of the first protein products that was designed with recombinant DNA technology in probably the 80s, 90s was tissue plasminogen activator.
Oh, yeah.
Which is a protease.
It cuts proteins. We talked about an HIV protease, right, in our first episode.
Absolutely. And actually, we talked about a strep A protease in the episode of that, cleaving our signaling proteins. So in this case, when you have a stroke, what's often happening is that a clot is lodged in your brain, and you want to break up that clot.
Mm-hmm.
And there aren't that great ways to do that chemically. The best we've got at the moment is TPA, which was these tissue plasminogen activators invented maybe 40 years ago.
Wait. That is a natural thing that's produced by our body, but it was produced in recombinant bacteria or yeast or something 40 years ago.
Yes, that occurs naturally, and the difference there was making it with recombinant DNA, so you can make it as a product scalably. Right. And so now imagine you could lay it on top some tweaking or some hallucinations, and you get even more useful proteases that perform similar or better functions. And, it's a tough problem because you also don't want it to run away and perform its function too well on the wrong strokes. So, but millions of people die of stroke every year. So it's those kind of targets just become way more tantalizing when you have more you can do with proteins.
Right. And I guess there are also other medical uses. So proteases are one, maybe other kinds of enzymes. Like there are a lot of rare genetic disorders where someone is missing an enzyme, or like some enzyme is dysfunctional or something like that. And then I guess there are other kind of diseases that occur where blocking a protein, or maybe like designing a new protein or introducing a protein or something, would help that person do some function that they weren't able to do before.
Yep.
And then would you also maybe be able to use proteins for diagnostics? Would you be able to use them for testing? Like I think I mentioned sometimes proteins change shape or something like that. If the temperature changes, I think you mentioned that as well. And then if the acidity changes or something like that.
Yeah.
And so maybe you would be able to make these really specific proteins that bind or go to a specific place. And then if something is there, it changes shape, and maybe that releases some information.
Yeah, you could design transistors with proteins. You could.
What?
I bet you.
I was not expecting that. What do you mean?
I mean, if all you're trying to do is send some signal under certain conditions and not others, you can make electronics with proteins.
Oh.
I assume. I'm making this up, but it must be true.
I don't know anything about engineering, so. Okay, so there are all of these really cool things that proteins could be doing, that people could be designing new proteins for. The like structures, the medicines, the diagnostics, the replacements for enzymes or hormones that people are missing, also the agricultural uses, or the like fermentation, or the industrial processes, or the materials, or
there's so many different things, and I think the starting gun is basically 2022, and what we're going to see, I believe, if people put the effort in, is a lot of structural biologists who know how to use these computational tools, but otherwise are essentially generalists, matching up with experts in particular fields who know a lot about diagnostics, or who know a lot about the heart, or who know a lot about a given infectious disease, or know a lot about a given agricultural problem. And in combination, I think those teams of people are going to do really incredible things.
I have a last question for you.
Hit me.
We talked about a lot of like applications of this and like making particular things. Is this going to be useful for basic research as well?
It's got to be. What is my first thought on?
Or I don't know, like understanding some disease or something like that, or understanding some process.
I mean, the answer's got to be yes, and then it's almost like start with the problem in hand before I know how to answer it. But the last 10 years, we had CRISPR come through. CRISPR has proven so useful as a basic research tool, maybe even above how useful it has been as a, medicine platform. So I wouldn't be surprised.
Yeah, I guess we talked about how, okay, if it can be used in diagnostics, that actually is a big research tool as well. Like definitely, if you're able to make sensors to something.
Yeah.
Okay, yeah, so there's lots of cool uses.
Lots of cool uses.
What's going to happen in the future?
There's two things I think we need to discuss, because we've just gone so far with these AI models. Number one is, are they going to cure us all of all diseases in the next couple of years? Sounds so magical. Why not?
No, I don't think so.
Okay, great. Well, I think we should get into that. And the other is, if you can hallucinate any protein for a function of interest, well, does that mean that terrorists can hallucinate proteins that attack other human beings?
Mm-hmm.
And we got to talk about that too. And that probably means a whole other episode.
Well, thank you for listening to our episode on protein design. And if you like this, share it with every single one of your friends, your family, your teachers, your haters, your colleagues, and subscribe.
Couldn't have said it better myself.